So now we've talked about two specific examples of two types of viruses. In particular, we've talked about double-stranded DNA viruses, which are considered group 1 viruses, and we've also talked about antisense single-stranded RNA viruses. And this is what should be highlighted, not the other one. Last year when I taught the class, I talked about HIV and RNA retroviruses. But the Ebola example was an example of a group 5 antisense single-stranded RNA. Now what I have listed all of these other groups for is not for you to go and memorize them. I'm certainly not going to ask what type of virus is a group 3 virus, but the reason that I'm putting this down is I want you to get the sense that there are all sorts of different types of viruses and that they each have different mechanisms that facilitate the viral life cycle. But as you saw through the two examples that I gave, they follow a very similar scaffold for the life cycle. There are a lot of similarities, and a lot of the differences are actually in the details of how they do it. So now we're going to talk a little bit about virus structure, and in particular we're going to start talking about the capsids. And I want to give you a few examples of a few different types of capsids. And one of the most common types of capsids are the icosahedral ones. And these ones have 20 identical triangular faces. And it's it's actually really cool how viruses are able to put together their capsids, because even though the capsid is formed by many faces, so in this particular example here is a triangular face, and each face is made up of 3 different proteins. The cool thing is that each protein is identical. That is, to make these complicated structures, really they don't need to encode a whole lot of proteins. They're able to keep their genomes small and just reuse the same subunits over and over again to form these types of structures. And icosahedral capsids are very typical of many animal viruses. And here you see a herpes virus example, and this is looking from the top down. So that's looking from the five-fold view of the capsid. Now another type of capsid that you see is the filamentous capsid, and you'll recall that, or you'll notice that this actually looks really similar to the Ebola example that I gave. And indeed, Ebola has a filamentous capsid. And the filamentous capsid has what is called helical symmetry. That is, the proteins that make up the capsid, each protein is identical, but all of the components fit together in a helix. And it forms this long tube of protein, and the genome is coiled inside of it. And once again, the genome of the virus only needs to encode 1 or 2 different proteins to make this big capsid. Now, there are some examples of viruses that have what are called complex capsids. So instead of just having, like I showed you previously, 1 protein that encodes the entire capsid, some of the capsid contains multi-part structures. And a bacterial phage is a really good example of that. So this has a capsid head, which is very similar to the example I showed you a couple of slides ago. But it's interesting here, you have this neck or tail sheath, and it's helical, and then you've got these tail fibers and a collar. So these are a little bit more complex, and as you can imagine, this adds to the number of genes that a virus must encode in order to form the capsid. And if you look at the genome of phage T4, of which this is an example, it actually is quite a large genome compared to the example I gave you of the Ebola virus. What's kind of interesting is that there are asymmetrical or irregular shaped capsids also. And the viruses that tend to have these irregular shapes tend to be larger viruses. And you might have seen actually in the past 6 months, there was this kind of cool discovery. They discovered this enormous virus in the permafrost that infects algae. And that had an asymmetrical capsid shape. And these tend to have DNA, and they're enclosed by a core envelope. They have lots of accessory proteins. And there's a working hypothesis that these appear to have evolved from degenerate cells. So at one point, perhaps, these were organisms, these were considered bacteria, and they were considered to be an organism that was alive, and over time they lost so many genes and so many capabilities that they kind of became a virus. So pretty interesting area of study, and I think that as we generate the capacity to sequence DNA of lots of bacteria and viruses, that this type of question will become more clear and that we'll be able to answer what exactly is going on with these large viruses. Just for fun, I'm showing you a couple of pictures of some viruses. In the first one, I'm showing you HIV on a white blood cell. And on the next one, I'm showing you a bacteriophage. So this is a bacterial cell that are covered by these phage particles. So you can see that they're all over the place. And they're kind of neat looking. Now, one of the questions is, how do you kill these suckers? How do you apply antiviral agents in order to get rid of infections? And one of the problems is that antiviral agents are really difficult to find. And that makes sense in the context of what we talked about as requirements for antibiotics. So recall that a lot of the targets of antibiotics were enzymes and cell structures that bacteria have that we don't have. The problem is that viruses use host enzymes. They use our ribosomes, or if they're infecting plants, they use the plant ribosomes, whatever. So they use our ribosomes, they often use our polymerases, they use our cellular machinery. And so there are a few targets that are actually unique to the virus. Remember that Ebola only has 7 genes. There aren't a whole lot of things in there that you can target. And if you recall too, that's one of the reasons why UV does a pretty poor job of killing viruses, is because UV damages DNA, and viruses don't have a whole lot of DNA to damage, so it takes quite a bit of UV exposure to kill a virus. The drugs that do work, so antiviral agents, the ones that do work often inhibit viral attachment. So this is one of the areas that the first step is the virus getting into the cell. So if you can prevent that initial step, even if there's exposure to the virus, you can either prevent or you can attenuate the virus from replicating. And I just have a couple of examples right here. You have this drug that inhibits viral uncoating, so the influenza virus can't uncoat, and because of that it can't get into the host cell. And this other compound, it actually blocks viral enzymes that degrades our mucus. And so what happens is that prevents the release of mature viruses. So there are a couple of strategies that can be used. There are a few more strategies that can be used by antiviral agents. And one of the strategies is inhibition of viral DNA and RNA synthesis. Now, when you inhibit viral DNA synthesis or RNA synthesis, oftentimes you're inhibiting the synthesis of the host DNA. This tends to still work because viruses reproduce so much faster than host cells that it affects the virus more than the host. Furthermore, if you're targeting, say, RNA-directed RNA polymerase, that is only found in viruses. It's only found in a subset of viruses, but that's one of the few things that, one of the few components of viral machinery that you can target. And I am going to skip these last two examples, I think. So those won't be on the exam. So I'm going to give you a little bit of a preview into the next lecture. What we're going to start into the next lecture is talking about bacteriophage life cycles. So we've focused a whole lot on the way that viruses affect us. You know, a lot of the study of viruses have been done with how they impact humans and also how they impact livestock and plants and things like that. But in the next lecture, we're going to move on to bacteriophage lifestyles. So that is that viruses that infect bacteria. And this turns out to be super useful in terms of genetic engineering for people studying microbiology. I think that I'm going to stop there, and I don't have any assigned reading for right now, especially because I deviated so much from the book. What I'll do is in the next lecture I'll come with the selection of some of the pages that you can read that corresponds to some of the lecture material. And when we start talking about the bacteriophage life cycles, we're going to start to transition back into book material. All right, have a good weekend.